Part 4 of my endomembrane project. Scenario D. Demonstrate the formation of a glycoprotein that would be displayed on the outer membrane of a cell for identification. So, like everything else, it starts with RNA in the nucleus being transcribed from DNA, and it leaves the nucleus through the pore complex and into the rough ER, where it will be first translated by a bound ribosome into a polypeptide, and then it moves through the rough ER cisternae, folding into its functional protein shape as it moves. Except this time, it's going to have a carbohydrate or a few carbohydrates added to it, because there are enzymes built into the ER membrane that add these carbohydrates to it, and that's what makes it a glycoprotein. So then it continues to move through, being folded as it goes, and then also ending up in the transitional ER, where it is again butted off into a transport vesicle. This transport vesicle again goes to the Golgi apparatus, where the vesicle membrane fuses with the Golgi and puts the protein into the Golgi apparatus to be modified as it moves from the cos to the trans side. And this time, as it is being modified, parts of the carbohydrates are removed. So the sugar monomers that make up the carbohydrate, some of them are removed and replaced or substituted with other monomers. So you can see some of the previously green sugar monomers on that have now been replaced with a grayish blue one. As the newly modified glycoprotein makes its way to the trans end of the Golgi apparatus, it is again butted off into a transport vesicle bound for the cell membrane. But this time, it's not excreted out of the cell. Instead, the membrane of the transport vesicle is fused and added into the cell membrane, and then the glycoprotein is embedded into the cell membrane. And then now it can be used for identification and recognition. And then I made some models of how these glycoproteins could be used by the cell. So here we have a cell with the red glycoproteins attached to its membrane and the green antigens surrounding it. So the green antigens can be detected by the glycoproteins, and the cell will know to commence phagocytosis and then destroy the antigen. Glycoproteins are also important in cell-to-cell recognition. So here we have a really simply drawn cell with glycoproteins on its membrane, meeting another simply drawn cell, also with glycoproteins on its membrane. They match glycoproteins. They recognize that they are individual cells and they do not digest each other. So um, everyone's happy. So there you go. There's my project. Thank you.